Okay, hi everyone.
Hi.
This is my sister Esther. She's going to help with the demonstration of protein synthesis.
Woo!
So, Esther, the two steps of protein synthesis are transcription and translation.
That's great. I don't know what that means.
You will.
Awesome.
Here we are in the nucleus. Come on over.
Woo!
All right, so I have here some DNA, 2 strands, and I want you guys to all notice that we're following the base pairing rule here. So on one strand, we have a T, and on the other side is an A, and then C goes with G. RNA polymerase is going to, your first job is to take these RNA nucleotides, which are floating about in the nucleus.
Oh.
This is Loki. He's going to help too by eating things probably. And your first step is to open up the DNA so that you can access the information on the inside. So I pull this up and you take that one down a little bit.
Does the RNA polymerase do this?
Yes.
Okay.
Oh, it's open. The DNA has been opened by the RNA polymerase. Now, let's see, which one are we going to do? This is the template strand. This strand starts at 3' and goes to 5'. And remember, 3' is with the little hydroxyl group on the end.
What is a hydroxyl group?
It's an oxygen and a hydrogen
Awesome.
put together. And then on the other end, we have the phosphate group, which is a phosphorus atom surrounded by oxygens. Template strand, 3' to 5'. Template is sometimes called antisense, and the other strand is the coding strand, which is the sense strand. But we're going to use the template strand. So RNA polymerase, G with C, A with T, start right here and go down.
Oh no. Okay, G with C, A with T. What is the purpose of this?
That's a great question. You can't quite do it like that though, because the RNA polymerase starts at one end and just goes right down.
Oops. Okay.
So the point of all this is to make an RNA copy of the DNA. And the RNA copy is going to take the information out to the rest of the cell, so the protein can be made. So the question is, why can't we... Oh, I see you're having a difficulty. There's no T.
There's no T's.
Oh, instead of T, we have U, uracil.
Why do we get to use U?
That's, in RNA there is no T, there's only U where T would be. And the reason for using RNA to go out into the nucleus instead of taking DNA out into the nucleus is because we want to have the DNA stay safe inside the nucleus. If the DNA is constantly going in and out, well, what if something happens to it? You need the original copy, you need to keep it safe. All right.
Can you tell me the purpose of this again? I already forgot.
So
I'm a great biology student.
the point of all all of this is to make a protein.
T, but there's no T. Okay. Oh, so I, as the RNA polymerase, make the protein by doing this process.
No, you do not make the protein. You make the RNA, and then another enzyme called the ribosome makes the protein.
So we're making RNA right now.
Yes.
Great.
And this particular kind of RNA is called mRNA. The M stands for messenger.
Okay.
There's 3 different kinds of RNA involved. This is the first one. Okay, so let's double check.
I don't think you have to do the second strand.
Awesome.
Nope, you don't. Because
Why not?
because that one doesn't have the correct information on it. The information is carried in the order of the letters. There's this problem though where RNA is not as stable as DNA. We're just making a temporary copy, but we've got to stabilize it somehow so that when it goes out into the nucleus, it can be used. So we put on a 5' cap.
Should I know what a 5' cap is?
It's just a molecule that goes on one end to stabilize it. It's like,
I hope you guys know that.
well, I mean, they should now.
Okay.
To stabilize it. So we're just putting that on there, a nice little cap, and let's put something on the other end too. It's called a poly-A tail. It's a bunch of A's, a bunch of adenosines. Play. Got this poly-A tail. There we go. All right, take this, zip that up. Okay, so then, RNA polymerase, thank you so much. You stay here.
Okay.
Except Esther, you can come be the ribosome.
Okay, so
All right, we're done. We're done with the nucleus. We've got
hello.
okay.
What to do?
So now we're going to take this RNA out of the nucleus through a nuclear pore, which should be here, but this is the nuclear membrane. All right, come on, Esther, let's go.
And this is the nuclear pore.
Yeah, all right, so now we're out in the cytoplasm and the rest of the cell. Let's straighten this out. Move you. So now we're in the cytoplasm, and I'm going to introduce you to the second kind of RNA that is involved in protein synthesis. Remember, the first kind is the mRNA, the messenger RNA. The second is the tRNA. The T stands for transfer. And what the tRNA does is it holds the amino acids and brings them to the mRNA. amino acids are necessary because amino acids make up proteins. And that's what we want to do, is make a protein. So we're going to need amino acids.
Makes sense.
But we also need them in the right order, which is where the order comes, well, obviously, the order comes in. This is what the three-dimensional shape of tRNA is. But for simplicity, we're just going to use this with the little amino acid at the top. Okay, so every single gene begins with the letters AUG. They always start with AUG. Groups of 3 on the mRNA are called codons because they each code for an amino acid. And on the tRNA, we have anticodons.
So you have to code the anticodons with the code on the mRNA. And that's what the ribosome does?
Yes.
Okay.
All right, so we've got all these tRNAs with their respective amino acids floating about. All right, now using the base pairing rules.
What are the base pairing rules?
Remember? A
Oh,
to
A
T,
with
or,
T.
except U in this case.
A with U?
Yeah, because we're an RNA.
Okay. Yeah, A with U, U with A, and G with C.
Exactly.
Okay.
Okay, so for the first one, we have AUG.
So I have to find one with UAC.
Perfect. All right, so the first protein,
Ribosome,
first
killing
amino acid is, ribosome, killing it, yeah.
The first amino acid is.
Methionine.
Will it always be that, or does
Yes.
it really depend on what the mRNA looks like?
No, it will always be methionine because the start codon is always AUG.
Right.
So we have AUG codon, UAC anticodon on our tRNA, which is the transfer RNA.
Okay, and then GUA to CAU.
Very good.
Then UCC to ACC to UGG.
All right, stop. Once you've got 3, you can start connecting them together. So the way it works in the cell is that there's spaces for 3 tRNAs inside the ribosome. It can hold 3 of them at once. But when it's time for the 4th tRNA to come, it takes this amino acid off, transfers it to the next amino acid, because it's transferring, and then out.
So I as the ribosome do that.
Yes.
Yeah. Okay, so I'm like carrying 3, and then when I have 3, I'm like, oh, I don't need this amino acid anymore. This, what is that called again?
tRNA.
The tRNA anymore, and I can take the amino acid and put it here.
Right.
Okay, and so I get a 3rd one, a new 3rd one. Okay, and then do it again. This is a C, right?
Yes.
Did it end on C? No, it ended on G. So then I need a C. So this one goes here. Oh, what do I do now? Because there's 2 there. Do I take both of these 2?
Yes.
Put them on here? Okay,
And the chain grows.
cool. And
There's
then we
a-
kick that one out.
There's a little Velcro on the other side of that.
Okay, and then this one goes here, and then I take all of these off. Okay, so this is essentially the protein becoming larger now.
Yes.
Nice.
At this point, it's called a polypeptide. A polypeptide is just a chain of amino acids. Once it folds up, then it will be
Oh, wait.
a functional protein.
So we get rid of that, and then we put this one here.
Yeah.
And then take all of these and put them onto that one and get rid of this, because I can only hold 3 at once.
Right.
Awesome.
And now there's no more. So you just finish up the polypeptide chain.
So I put it over here.
Yes.
Where's the amino acid on this one?
An excellent question. There is no amino acid on that one. This ACU corresponds with UGA, and that is a stop codon. The stop codon means the protein is over, and it doesn't carry any amino acid. it just stuffs itself into the ribosome, pushes everything over, and then just ejects right back out the way it came.
So this is the stop codon?
That's the release factor. The
Okay.
stop codon, remember the codons are the groups of 3 letters.
Right. And the stop codon is the last one?
Yeah, this one, UGA.
Okay, why isn't that the... Oh, wait, it ended there, and then this is the A thing. What is that one
The
called
poly
again?
A tail.
The poly A tail.
Right.
Right.
Good.
Cool. So now...
Oh, did the Velcro not work on that one?
No, I just
Oh.
did it the wrong way. There we go.
Take the release factor out as well.
That's
Yeah.
the release factor?
There we
That's
go.
the release factor.
Okay, so now we have our polypeptide, and it's going to fold up into a 3-dimensional shape. There we go.
Oh.
And now it's a protein.
Okay, how does it fold up into the 3-dimensional shape? It just does?
Great question. So what it does is, see these little groups on the sides, on the bottom of the amino acids. Each one is different, and they have different properties. Then that one is going to fold outwards towards the water.
Okay.
And then, because that's going outwards, maybe it, let's see, tryptophan. Ooh, that's pretty big. So this one would fold, let's say that's hydrophobic. So it goes to the inside of, let's say that's hydrophobic. So it would go to the inside of the protein to be protected from the water. But it's awfully big, so it's going to take up a lot of space in the middle there. And then this one, oh, that's super small. That's hydrophobic as well. So there we go. Do you get the idea?
Yeah. Are they literally called hydrophobic?
Yeah, hydrophobic.
That's funny.
Hydrophobic just means water hating.
Yeah.
Yeah. Oil is hydrophobic.
Okay. Makes sense.
The last thing, what happens to this and what happens to these?
I have no idea.
That's
What
a
happens
great
to
question.
them?
So the empty tRNAs go back to a different enzyme that will put more amino acids on them. Always the same amino acids. So this one is going to have another
Makes sense.
threonine on it. Just gonna go get another threonine and then be able to transfer again.
Awesome. That's very
et cetera.
sustainable of them.
Yeah, it is very sustainable.
Yeah.
That's a good point. The mRNA can be used sometimes again and again. Sometimes it will last for a little while. Sometimes it degrades right away. There we go. That, my friends, is transcription and translation.
Awesome.
Thank you, ribosome.
You're welcome.
Okay.
So in summary, these are the two, I guess, strands of DNA, and as the RNA polymerase,
Polymerase.
I open this up, and then the mRNA comes and bonds with all of the right parts of this by the bonding factor thing that we learned. Is that what it's called? Like the rule?
Oh, yes,
G with T, C with...
the base pairing rule.
Right, the base pairing rule.
So the RNA polymerase is the one that puts them in the right order.
Yes.
Right.
And then the C cap goes on, and then
5' cap.
the 5' cap goes on, and then all the A's then
The poly A tail.
poly A tail.
Very good.
And
what's the point of those?
I don't know.
To keep the RNA from degrading because it's not very stable.
Okay.
To make it last longer.
Yes.
Because it's got to last at least a little while longer.
So then it all goes on there, and what happens after that?
Okay, well, first of all, this is the process of transcription. That's
Right.
transcription. All right. Then we take our mRNA. The DNA goes back together.
And the RNA stays there.
Polymerase, yeah.
Polymerase.
Woo! Out of
And
the
then we go through the
nucleus.
pore.
Yes, out of the nucleus into the cytoplasm.
Woo! And then the ribosome, the mRNA is chilling out here.
Yes.
And this is what translation is.
Mm-hmm.
And so all of these things, what are these called?
tRNAs.
The tRNAs, which already have amino acids on them,
Right.
go and pair with the mRNAs,
Right.
and then every time there's 3, because the ribosome can only have 3 of them at a time, so then the amino acid, transfers over to the next T.
It attaches to the next amino acid.
Next
Yeah.
amino acid, until it's done that all the way up, and then you have your protein.
Right. And do you remember what a codon and an anticodon are?
No.
Okay. A codon is a group of 3.
Right. Yeah, and the codons have to match with the anticodons, which are here.
Yes. Very
Right.
good. Very good. So each codon codes for one amino acid, and it is the order of the codons that determines which protein will be made because of
Okay.
which amino acids are used. In what order?
So it's essentially the order of the mRNA that determines what protein will be made.
Right.
Also.
And what determines the order of the mRNA?
The order of the DNA.
Exactly.
Cool.
So,
Oh.
if you go into a cell and look at the DNA, which we can do and find the DNA sequence of something, then you know what the protein is going to look like.
Why is it important to know what the protein is going to look like?
Well, you know what protein is going to be made.
Okay.
I guess you may not know its physical appearance, but you know which protein is going to be made. And it's just important because it's important for things like gene therapy, for example. If you can identify a gene that is damaged, it's going to make a damaged protein. The protein won't be right. And so maybe you can go in and correct that
Awesome.
by delivering a functional copy of the gene to the cell.
Nice.
Okay, in summary, the process of transcription and translation together are called protein synthesis because you're making a protein. Transcription happens in the nucleus where the RNA polymerase takes apart the DNA and using the base pairing rules, A with T, U because it's RNA, G with C, correctly lines up the nucleotides of RNA. That's why it's RNA polymerase. Then, because RNA is not very stable and we need to keep it stable, you add a 5' cap and a poly A tail. Poly meaning many, A for adenosine, which is A. 5' cap and a poly A tail. All right, DNA snaps back together. Out we go to the cytoplasm.
But the RNA stays there.
Yeah.
And then this is translation.
Yeah, so you can remember it by transcription is like transcribing. If you're writing something down, you're writing down the DNA into an RNA form.
Okay.
And then translation is when you're translating it into a protein. All right, so here we are at translation. The ribosome recognizes the start codon. AUG is always start. Each group of 3 is called a codon, and each group of 3 matches with a specific anticodon on a tRNA, T for transfer, because they transfer amino acids from themselves to the protein. And then, when the codon and the anticodon match, the ribosome continues matching until there's 3 tRNAs inside the ribosome, because it can only hold 3. So at that point, the 4th tRNA comes in, the first tRNA leaves, and then this amino acid is attached to the next ones.
And that keeps going until until we get to the end of the mRNA, and then all of the amino acids are together, and
then the release factor sticks in. There's no amino acid on the release factor. Sticks in. Bye, guys. Those guys, the tRNAs, will go to have more amino acids put on them so they can be used again. And then we have our protein.
and then it twists together into a 3D shape.
Right. And the 3D shape is the protein. Great. I should have said, and then we have our polypeptide. I should have said that.
Awesome.
Polypeptide protein.
Awesome.
Thank you so much, Esther, for helping.
You're welcome. Thank you.
All right, now you can go tell all your friends how cool protein synthesis
No,
is.
it's really cool though.
Isn't it?
Actually, it is. Yeah. Yeah. Cool